There you go.
And here's genetics review. Okay, I can't look at it too much. All right, genetics, the study of heredity. I want to first review the structure of DNA. DNA is a complex molecule. How many different strands of smaller molecules are in a DNA molecule? Raise your hands. How many strands in a DNA molecule? You guys, I'm not going to sit here and watch you guys not use your notebooks, not look for this information. You need to help me out. 2. DNA, you have 2 strands. They are twisted, but I'm going to draw them sort of flattened out. If I draw it this way, this is considered the backbone of the molecule, and these are the rungs of the ladder that are attached by hydrogen bonds. What is the backbone made of? Raise your hand.
Phosphate.
And sugar. Don't forget, your nucleotide, which is the monomer, has a phosphate group, it has a sugar, and it has a nitrogenous base. This is your sugar phosphate backbone. So that means these rungs of the ladder are your nitrogenous bases. They're just smaller molecules that have a lot of nitrogen in them. What 4 nitrogenous bases can you have in DNA? Raise your hands. Good, good. Yep. Guanine. So adenine, thymine, cytosine, and guanine. These can be the different bases in DNA. If I have adenine here, what does it always bond to? Raise your hand.
Timing.
It is always with thymine. If I have a cytosine, what is it always bonded to? Guanine. All right, so if I fill in some more of this sequence, you can tell me what the complementary strand is. Beautiful. Now, one thing we talked about in my last class that hopefully is on YouTube for you guys is DNA synthesis, part of the cell cycle. You need to make sure you review that. I do think it's worth pointing out here for the second class in a row, however, that during DNA synthesis, MCAS loves to ask this. Way back when in the beginning of the year, a lot of you lost points on an open response question about this. They love for you to point out the fact that in DNA synthesis, what do I mean by DNA synthesis? Raise your hands.
When DNA is being made.
Say it again.
When it's being made.
Yeah, when it's being replicated. So you're taking all of your DNA and you're making a copy of it. You have to realize that during DNA synthesis, each strand is a template for the new molecule. So at the end of DNA synthesis, you're going to have 2 molecules exactly like this. Each one will be made up of an old strand and a new strand, an old strand and a new strand. You have to realize that about DNA synthesis, okay? If you don't know what the heck I'm talking about, I did a very thorough job explaining it in my last class's review. Again, that's why I'm putting this stuff on It's Learning and YouTube for you, all right? All right, good. So your nitrogen, this is a hydrogen bond, they're not chemically bonded, okay? Questions about structure of DNA? I'm gonna shut this just until the class.
Can I go to the bubbler?
Yeah, just shut it when you come back in, okay? All right. Now we could talk a lot more about DNA, but we have many other things to talk about in genetics, so we're gonna hold off on that for now. Actually, you know what? Turn your page over. I want to do a transcription and translation first, and then we'll go backwards and talk about meiosis, okay? Transcription and translation is considered, I'm going to keep this up here, it's considered the central dogma of biology. I would definitely remember the basics first and then try to work through some of those details. Transcription and translation. Somebody tell me very basically what's going on in each of these 2 processes.
Transcription is when DNA is transferred to RNA, and translation is when RNA is transferred to proteins.
Good. So DNA, specifically to messenger RNA, is your transcription, and your translation would be using that RNA to make a protein. I mean, this is must know stuff here. It wouldn't be called the central dogma of biology if it wasn't really important, okay? Couple of tricks to help you remember which is which. Are DNA and RNA the same type of molecule? Yes or no? Raise your hand. DNA and RNA, are they the same type of molecule?
Yes.
Yes, what type of molecule are they?
DNA.
What's the type of organic molecule? Nucleic acid. DNA and RNA, both nucleic acids. Same language, just slightly different, okay? Proteins, what are proteins made out of? Amino acids, not nucleotides. They are not nucleic acids. It's a completely different language, a completely different molecule, okay? So starting with how to memorize this, in translation, you're translating from a nucleotide language to an amino acid language, totally different, okay? Now as far as transcription, ignore all of biology for a second. If I said to you, a few years from now you're going to apply to college, and I'm going to say to you, I need a copy of your high school transcripts. What does that mean? What is a high school transcript?
Grades.
It's a copy of your grades. It's still your grades, but it's not like you're picking up Aspen and saying, here are my grades. You're giving them a copy of your grades. Your transcript is a copy of your grades. Your transcript is a copy of your gene. Okay, so transcription is copying your gene onto RNA. Question so far? All right, there are a couple of details you do need to know about this. First detail, if this is in your nucleus, why is this in your nucleus? Kind of a silly question, but basic gen bio here. Why am I putting that in the nucleus? That is where the DNA is stored. In what kind of cells? In eukaryotic cells, true. All right, so if that's in the nucleus, way out here in the cytoplasm, you have ribosomes. Again, cell biology review is last period, but we have to talk about it for transcription and translation. What does a ribosome do? Raise your hand.
Makes protein.
The ribosome makes the proteins. If your DNA is the code to make the protein, and the machinery to build the protein is out in the cytoplasm, the reason why we have messenger RNA is to get that gene, those instructions for building the protein from the nucleus to the cytoplasm, okay? So the very first thing that happens when your cell needs to make a protein, a copy of your gene is made, but not onto a DNA molecule, onto an RNA molecule. So here is your messenger RNA. If the messenger RNA is going to be built to be a complement to this strand, somebody raise your hand and tell me the sequence. Good. Don't forget, in RNA it's U and not T.
G.
Yep.
G.
Yep.
U.
Yep.
A.
Good. What is this process called? Raise your hand.
Transcription.
This is your transcription, making the messenger RNA from the DNA. Then your transcript leaves the nucleus, comes out here. The ribosome actually clamps onto it. What is your ribosome made out of? What actually makes up a ribosome?
Proteins.
Say it again?
Proteins.
Proteins, true, and what else? Type of RNA?
tRNA.
Not tRNA.
Ribosomal.
rRNA, ribosomal RNA, okay? So don't forget you have different kinds of RNA and you have to know what each one does. Your messenger RNA is actually the gene itself copied onto an RNA molecule. Dan, what's up? Put that away. Your ribosome is made up of your rRNA. So the only guy I haven't really talked about here is your tRNA. What is the role of tRNA in this whole process? Look it up in your book if you don't remember, you guys. I asked you to bring your old notebooks. You have textbooks in front of you. Somebody raise your hand and tell me the role of transfer RNA. Just because you have your phone out doesn't mean I'm going to forgive you just for answering the question.
Okay.
It's my evil eye
Put it in my
trying to
hand.
be off screen. Tell me the role of tRNA.
It transfers It transfers it from the nucleus to the fat.
Not a bad guess, but not quite.
It transfers it from messenger RNA to an amino acid.
You're close, but you're not putting it totally correctly.
It attaches to the amino acids and the messenger RNA needs to build the protein and attaches to the messenger RNA and joins the amino acids together.
Yeah, the only thing you said wrong was it's actually the ribosome that builds the protein. The ribosome catalyzes the reaction to connect amino acids. But what you said is correct. Your transfer RNA on one end has an RNA sequence, so it might be, let's say, UUA. And on the other end, and you can use a chart, the genetic code, we did a lot of work with genetic code, you have one in your book. The other end of it has a specific amino acid. Now if that's UUA, and there's a part on the messenger RNA that's complementary to that, so it would be AAU, that is the signal for this transfer RNA to come right over to the ribosome and bring that amino acid to the growing peptide chain. You have to make sure you know the role of your transfer RNA. It brings one amino acid at a time, the correct amino acid to the ribosome so the ribosome can connect them to build your protein, okay? What I usually see for questions on MCAS will be pure what is transcription, what is translation type of questions. MCAS, however, does love to show a picture of part of these processes and asks you to identify what process it is. If you see a ribosome, a messenger RNA, and a transfer RNA, you should know right away that it's translation, going from your nucleotide language to your amino acid language. What does polymerase do? If I told you somewhere in this whole entire set of processes, you had something called RNA polymerase. Where is that important? Raise your hand.
It's important for transcription. It's like the process of looks like when the DNA is being switched to the RNA or copied to the RNA.
Yeah, there's actually an enzyme that takes one nucleotide at a time and builds the RNA molecule based on the DNA strands. That enzyme is RNA polymerase. All right, good. And the only other question I have for you, which one's the codon and which one's the anti-codon? Take a guess.
Is the codon like the 3 that's like on the MR?
Yes, yes. This is the codon. This is actually the code saying what amino acid goes next. The anti-codon is the sequence that's on the transfer RNA molecule. It's anti, it's opposite, it's complementary to the code on the messenger RNA. And again, your anti-codon is always paired with a specific amino acid. All right? Really good. Any questions about transcription translation? Anybody totally sick of biology class yet? You got that on film. Of course, we also got me almost throwing something at her on film too. All right. Can I get one more section out of you guys before you have your break? Did you say yes?
Yeah.
Thank you. All right. Transcription translation is done. Go back to your previous page. All right. Now, everybody's gone back to meiosis. We're going to talk about meiosis as quick as we can, and then I'll let you guys take a quick break, and I'd like to finish up with like Mendelian genetics, okay? It's tough for me to jump into meiosis because I did not review mitosis with you. However, that's what I did in my last class, and it's going to be on YouTube for you guys to check out. I do want to point something out now, though, before we talk about meiosis. Somebody in very simple terms. Tell me the difference between these two.
Mitosis breaks it up twice, and meiosis breaks it up. Or mitosis breaks it up once. Meiosis breaks it up
What
twice.
do you mean by it?
Well, it splits it in half.
What's it?
The cell.
The cells, specifically the chromosomes, alright? Give me some more.
Mitosis is asexual reproduction, and meiosis is asexual reproduction for the purpose of sexual reproduction.
We're gonna say that this, gets the cells ready for sexual reproduction, true. What else? Somebody tell me something else.
Mitosis forms two genetically identical diploid cells, and meiosis forms 4 haploid cells that are all different.
Beautiful. Alright, so mitosis, which is asexual reproduction, you end with 2 identical cells that are usually all diploid. Meiosis is setting up for sexual reproduction. At the end of meiosis, you have 4 totally different cells and they're haploid. What do I mean by diploid and haploid?
Haploid is like half a cell, half chromosome.
Haploid is half the number of chromosomes. Diploid is the normal number of chromosomes. Diploid, 2. Why does diploid have the prefix 2? I mean, DI stands for 2.
It has both sets of chromosomes.
Where do you get them?
Each pair.
Yeah. In a diploid cell, you have a complete set of chromosomes, which means you actually have pairs of chromosomes. Diploid means you have both sets, one from your mother, one from your father. A haploid cell only has half the number of chromosomes. And that makes sense because in sexual reproduction, when you take a cell with half the number of chromosomes and you combine it with a cell with half the number of chromosomes, what you get is a cell with the complete number of chromosomes. Okay? What is the diploid number for humans? Raise your hand. Annalise?
46.
True. What's another more accurate way to say that?
2N.
2N, true. What's another? 23 pairs, true. 23, 23 pairs. I might delete the whole entire recording before I post it. I'm telling you, it's gonna be embarrassing. Okay, 46 in humans, that means 23 pairs, a whole set from your mom, a whole set from your dad, and that is your 2N number. So what's the haploid number in humans? In other words, I know this is awkward, but in all of your sex cells and my own sex cells, my egg cells, your sperm cells, whatever, how many chromosomes? Raise your hand. Haploid number for humans. 23. And again, and that would be considered 1N, or just N. And that makes sense, because when you have fertilization, 23 plus 23 is going to bring you back to your proper chromosome number, okay? Must know. We have to go into a little more detail about meiosis. Can I erase this? You guys good? There's somebody still writing?
You have to write this one.
Hang in there, you guys. We have a break coming in a couple more minutes. Be appropriate. You're on film.
I'm actually not on film, it's just my voice.
So you're on film.
Just tell me if I need to edit anything they say out.
Yeah.
Oh, alright. Alright, here we go. Meiosis. I have to stress something now that again I covered in the last class, but I have to mention it now. It doesn't matter if it's meiosis or mitosis. The first step in the cell cycle is the same, okay? So without going into too many details about the cell cycle, what is the first half of the cell cycle?
Grow and develop.
Yeah, what is that called? Like the whole thing?
Prophase.
Nope, that's part of mitosis.
Interphase.
Interphase. And again, you have G1, you can get all this detail from the other set of notes, G2, you have S phase in between. This is the same whether the cell is going to go through mitosis or meiosis. The cell still needs to get ready for DNA synthesis. The cell is still going to copy all of the DNA, and the cell is still going to get ready for division. It's this part, color, It's this part that is different in mitosis and meiosis. Really quickly, somebody remind me the stages in mitosis.
Mitosis.
Yeah.
Is it prophase, metaphase, anaphase, and
Telophase. Prophase, metaphase, anaphase, telophase. Please make another taco. And I can say that now because I ate my lunch and I'm not hungry anymore. One set of division in mitosis. In meiosis, these stages happen twice. The first time, you separate your homologs, and the second time, you separate your sister chromatids. What is the difference between homologs and chromatids? All right, let's do this. If I show you a chromosome like this, and I show you a chromosome like this, and then I show you a chromosome like this, what is different between this chromosome and these two chromosomes?
That's,
they were separated. So they're like when they're separated, they're the sister chromatids.
Sort of. What's the matter? Are we losing me?
No, we're zooming in on your picture.
Boy, I should have done a better picture if I knew that. No, just kidding. All right, sort of. Help her out. What were you gonna say?
that is a one total chromosome, and those are two separate homologous chromosomes.
What are you calling homologous? This or this?
Right.
These are homologs. One from mom, one from dad.
Great.
These are not homologs. This is one chromosome copied after S phase. This is the copy. This is not chromosome from mom, chromosome from dad. This is one chromosome from mom. In order for me to show you homologs, homologous chromosomes that have already been copied, I would show now these are homologs, a set from mom, a set from dad. This is the copy of the chromosome from mom. This is the copy of the chromosome from dad. There's a lot of stuff that goes on in meiosis 1. What happens in meiosis 1 before you separate your homologs? What is the big important thing that happens in meiosis 1?
Crossing over.
What does that mean?
It's when the DNA, or not the DNA, the chromosomes, switch pieces,
True. Are you switching pieces between sister chromatids or between homologs?
homologs.
Yeah. During prophase 1 of meiosis 1, you have a piece of the maternal DNA switching places with a piece of the paternal DNA. These are homologs, one from mom, one from dad. Metaphase 1, they line up at the center of the cell. Anaphase 1, your homologs separate independently of each other. So you might have paternal over here and maternal over here, but the next set of chromosomes could be the opposite. They're not related. In meiosis 2, you're separating your copies. You're separating your sister chromatids. So meiosis 2 is actually more like mitosis, because that's when you're separating the copies, okay? Make sure you realize there are 2 rounds of division in meiosis, and also realize that's where crossing over happens. That's why every single gamete is completely different. All right, now before I tell you I am done with meiosis, somebody tell me what a gamete is. Okay, everybody's hand should have flown right up. Who needs the point here? You get a point for helping me with the camera. Go ahead. Your gametes are your sex cells, your egg cells and your sperm cells. So are they haploid or diploid? Haploid. That is the cell that gets made from the process of meiosis. Okay? Any questions or comments? Is anybody dying to have a break?
Yeah.
All right, you guys, if you want to pause it, I'll give you 5 minutes, but when I get